All right, we are recording. So welcome everybody to this month's StaPH-B call. I think that we've got an exciting call ahead today. I am looking forward to it very much. I guess just a little bit of a background. For those of you who don't know, this is StaPH-B. So this is a group of self-aggregated bioinformaticians across the states and the federal government and beyond now. I think we've got some other people now from industry and from all sorts of different kind of sectors. So this is really exciting. We started a couple of years ago, but we've grown tremendously in the past couple of years. And I think that we are now somewhere in the 150 range in our group membership. So it's really exciting to see how much this group has expanded and grown in the short time that it has been around. So today's call is going to be focusing on CLIA CLIA validation and CLIA experiences for sequencing, next generation sequencing. Last month, Varvara contacted me and said, hey, I have a great idea for a call. Would you, do you think that this is a good idea for a call to have happen? And I thought that it was perfect timing because a lot of people were just starting to kind of see the variants that were kind of exploding at that point, and a lot of people were just starting to kind of talk about the potential implications of sequencing and how do we report on sequencing for SARS-CoV-2. And so this has kind of been something that has been percolating for a little while, and it's really coming to a head now, I think. And so I think that this is a really timely call in order to have, just because a lot of people are facing kind of these same issues. And so we put together a small little panel to kind of answer some questions about experiences with CLIA regulators and CLIA validation. And then I also think that if we have time, we've got kind of an ambitious agenda today, but if we have time, I would really like to open this up and kind of, capitalize on the groupthink and the hive mind here for a lot of this discussion, and kind of throw it out to the audience and really include a lot of people in this discussion, because I think that there's a lot of great minds on the call, and we can kind of come to some answers together on some of these issues. So to start it off, do you want me to introduce our panelists, Barbara, or? Okay. So I just invited a couple of people who I knew had some experience with CLIA and sequencing before. And so Nicholas Matlock from Maine was invited to be on the panel, as well as Kelly Okeson in Utah. We've got Ritu and Matt from California. And we also have Marianne Turnsick from CDC, providing kind of the CDC perspective in some of these areas. And so with that, I think I'm going to turn it over to Varvara, our moderator, who has come up with a list of great questions, and I'm excited to hear what people have to say. So with that, I'll stop talking and turn it over to you, Varvara.
Thank you, Logan. And just to add a few notes to what you have just mentioned, that our thought was to put an emphasis on the actual CLIA inspection experience that labs have had by now, and any details are appreciated. For example, if you could please mention whether CLIA inspector actually looked at the specific parts of your quality management system or the validation and was okay with it or not, please share, it will be very helpful. And even though we have our esteemed panel, our plan was to have a rather unstructured conversation. And so please pick up on if you have any input on the questions that will be presented on the slide. And even if you have the same experience with your CLIA inspector that already was mentioned by some other lab, still please share it. I think it might be helpful for us to understand what might be just one inspector's opinion versus a more consistent approach from the CMS. But then, of course, we are welcoming any thoughts and feedback and questions that you might have on the topic. And well, as we all know it, there's more than one way to skin a cat, or if you will, there's more one way to CLIA. So without further ado here, the questions that we posed to our panelists. And we identified these 6 main topics that we would like to cover today. And they're all here on the slide, but if also time permits, there's some additional questions here that we hope to address in the end of the call. So our first question is, what is your experience in CLIA validation of next generation sequencing testing? If you could please cover whether you have gotten any feedback from your CLIA inspector during the inspection on your validation approach. Did CLIA inspector question any part of your validation, and what was your response? What was the number of samples that you selected for validation, the actual samples that were sequenced versus in silico samples? What were your true positives versus true negative validation samples, or anything else that you would like to address in your CLIA validation experience? And first, I would like to hear from our panel members, if anybody would like to share their experience with the validation.
Well, I guess I can start off. So we have three tests now under CLIA that use NGS. We have a Salmonella serotyping, an E. coli serotyping, and then as of about a week ago, we have our Illumina COVID-seq under CLIA as well. And we just had our inspection, just ended yesterday, actually. So this is my week of CLIA, I guess. So 2 years ago, during the inspection, when we first had our Salmonella serotype plan validated, we did get some feedback from our CLIA inspectors. We basically showed them the validation plan. They had some questions about things, but the questions were, how do I put this nicely? Not really on topic. They didn't really understand the NGS system or the approach or what we were doing. And they were asking all kinds of weird, crazy questions that didn't really make any sense. So we basically took the approach then to basically put together a little presentation and you know, give them a class on what NGS is and how we're using it. So really the feedback we got during that inspection was, I mean, it was questions like, how do you know base space isn't modifying your data? You know, some really kind of weird, bizarre, out there questions that just, I think, went to, you know, speak to how they didn't understand the NGS or WGS process. They read the validation plan, didn't have any real major concerns or problems with it for any of those things, for any of the processes we had in place. Let's see what else is on here. I think, again, that goes to the fact they didn't understand what they were looking at in the first place. For our validation, we did for Salmonella, I want to say it was around 300 or so samples that we did compared back to molecular serotyping. And it was about I don't know, 3/4 of those were positives. Or actually, no, I'm sorry, I take that back. Most of those were positives, and we threw in some contaminations as well. So we threw in some E. coli, some Salmonella, or some Campylobacter, a handful of other PulseNet bugs that we would be sequencing on the same run just to make sure we could detect those. Did the same thing for our E. coli's. And then as for our COVID-seq, we also did, I think it was close to 1,000 samples for that validation.
And all of those were actually sequenced, or that's in silico samples?
All those were sequenced, yeah. Because basically we had, you know, 2 years worth of PulseNet bugs that we had sequenced, and that had also been serotyped with a molecular assay. And so we just did a quick comparison to say, yep, what we sequenced and what we called in silico met up and matched, you know, 99% with what the molecular assay said.
Okay, makes sense. So it means that you just got the historical data, which was generated by your lab, but you not necessarily had to sequence 1,000 samples after the moment that you submitted the validation plan to your lab director.
For COVID-seq, yes, we did have to sequence all of those samples. For Salmonella and E. coli, no, we used historical data, and then did some new sequencing for our, you know, between run, in run, between user, between day components of the validation. But those were a much smaller subset of the total. I mean, it was like 30, I think.
Kelly, for your Salmonella and E. coli, do you use bionumerics or do you use command line or both for the CLIA workflow?
For the CLIA workflow, we actually have a pipeline we've created here in-house that is an automated pipeline that uses Seq0 and serotype finder for Salmonella and E. coli respectively. So we had this done before bionumerics had even updated to the new version for whole genome sequencing.
Okay.
Yeah, so this is Nick from Maine. We sort of had a similar experience. So we don't use bionumerics for any CLIA work because I have no control over any of the workflows. So we'll never validate it at all. So we use CLC Bio a lot. And we built one large program for all the PulseNet organisms and all the HAI bacteria. So independent whether it's Salmonella or E. coli or Vibrio or Staph aureus, I'm going to run that through our entire workflow. It's kind of labor intensive on the in silico side, but whatever. I still get all the results out. So we probably validated close to 300 to 400 different PulseNet organisms and over 500 HAI organisms through that workflow. We look for, what do we validate? Vibrio, serotype, Salmonella, E. coli, MLST, strep pneumo, paratype B, virulence factors, and antibiotic resistance genes. We've had this going for about 5 years now. CLIA inspected it maybe 2 years ago. And the way we approached it was we did IQCP, which you're allowed to do. And we broke it down to every individual question we could think of that CLIA would ask. And we said, before you start asking questions, I'm going to go down this massive list of questions and answers that I think you're going to have. And I took them step by step in the process. I click on this button, it does this. These are the type of risks we look at, yes or no. Does it affect the data, yes or no? We even did IQCP to not have to run controls for a lot of our stuff, which they found was really, really interesting that we were able to get around it. We did CLIA validate metagenomics for CSF, and we do have controls for that. We do have an RNA and a DNA phage for extraction and PR all the way through. And we just very, very specifically set, here's a table of all the metrics we look at the beginning that we have to hit to make sure we're not, so we don't have to use a control, because sometimes, a specific metric can fail, the rest of the run is fine. So we just spelled that out front. And they really like to see, they want to know, well, since it's software, what happens if you upgrade the software? And we have it pretty well spelled out when we do and do not have to do a new validation. Like if you change the color from blue to green, I don't care. No one cares. If you change where a button is placed, I don't care. But if you fundamentally change an assembler, I might have to do a smaller validation.
I can talk a little bit about what we did within EDLB for CLIA validation. Hi, everyone. This is Mary Ann Turnsick. I'm part of the Enteric Diseases Laboratory branch at the CDC, which is home of PulseNet. So we first did a validation for ANI in bionumerics, and we included about somewhere over 300 strains in that original validation plan that we did. And that was for 3 organisms only: Campylobacter, 6 Campylobacter species. It had 6 Listeria species, and then 3-4 Escherichia. I'm blanking on that. But for those 3 taxons, we had approximately, over 300. And we have since changed the ANI tests. We've added the Salmonella and Vibrio to the test. So we went through the revalidation and we did that piecemeal. The first thing we did is we updated the reference genomes. We added the additional genomes to the test, and we call our reference genome data set the RGD, reference genome data set. So we are now on RGDB2 within binumerics for the ANI test. So once we added those new genomes to the RGD, we reran our original validation data from those 3 taxons, the Campylobacter, the Salmonella, and the Escherichia. And we showed that those additional genomes did not, that change did not affect the outcome for those original assay targets. And then once we validated the new RGD, we validated the new species, the Campylobacter and the Vibrio. And then for that set, we probably had about maybe 60 or 70 different representative species. For our true negatives, we use things that currently aren't identifiable by ANI because it's not in the RGD. So we use things like Klebsiella, Helicobacter, Yersinia, Chronobacter, but RGDB3 will contain Yersinia and Chronobacter. So they will no longer be able to serve as our true negatives in the test system. And we will follow the same sort of strategy. First, we'll validate RGDB3, and then we will do another validation for the Yersinia and the Chronobacter. And these are very sort of mini validations. They're not as comprehensive as the original ANI validation was. And again, that first part, when you change the RGD, that's just rerunning sequence data that you already have. So you're not going back to the DNA extraction phase for that. When we validated a new, when we made a wet lab chemistry change, validated a new DNA extraction kit, new library prep kit, then we do have to go back from the beginning and do those extractions and take it the whole way through to analysis. We've had a couple of, I think we've had 1 inspection so far. Our next inspection is at the end of April, so we'll probably have another, we'll talk about that some more during one of the 50 state calls, or maybe another office hours. I'm sure we'll talk about it again. So I'll keep you posted as to how that goes. But the first one was fairly smooth. They did ask some questions, and I would agree with Kelly that they weren't, those questions kind of gave away the game, that at that point, you know, this was what, 2 years ago now, they really weren't savvy to the technology. But I know there's a lot of conversation going on out there. So I am more curious as to what types of questions will be coming to us at the end of April.
And I can speak to my just, yesterday CLIA inspection. They didn't really seem to fully understand NGS or really even ask any questions other than where are your proficiencies? Where are your, that was about it.
Yeah. We do, because there's not, we do alternative assessments, to satisfy that requirement. And then if we get a organism in via one of the PT programs that we participate in, and it happens to be an ANI organism, we will use we can satisfy our WGS PT requirement with a CAP organism occasionally if we get one. They always give us something like an Aeromonas, and so, go back to good old RPOB. There was one other thing. Maybe that's all for now. Oh, I know what it was, and then this is really all for now. When we first did our, when we first implemented WGS within EDLB, we did not use external controls, but that was something we had to implement once we were already into the process, and it was a challenge for us to do that. We rejected it. I think that's the normal reaction, but I can say that we have found them to be very useful, external controls. We use them to do our instrument comparisons. We use them to check the integrity of the pipeline. They can be used for multiple things, so we do find them useful. But I'm with you. I can't remember the folks up in Maine. They did their IQCP. We have one going as well. We don't necessarily want to use them on every run. We would like to do what you guys are doing and use them periodically as we define it. And that's it.
I think
Hi,
there's a
everybody.
question to Marianne. I'm so sorry. Just to follow up, the question is, Marianne, you're using the sequence for the type strains of these species for the database, question mark. I may have missed that. Thank you.
Let me read. Sorry, I hate when I have to crawl up. I'm on my small laptop. You guys are just gonna see my wrinkles. Forget it.
Just a question about if you're using
You're
sequences
using the
of type
sequence for type
strain.
strains of these.
for database.
Well, if we have the type strain, we'll use it. That's always a good one to include in your data set. But there are a few holes in our collection. We might not have every type strain on hand, but we do, when they are available to us, we do include them.
Thank you. Yeah, okay. Go ahead, Rita.
Hi, can you all hear me?
Yes.
Yeah, thanks, Barbara and Logan. So yeah, this is Ritu from California, and we can share our experience with the CLIA inspection. We had so far 3 CLIA inspections. The first one was in 2016. So all 3 inspection were very different in our experience, like questions and the things they looked at every time it was very different. So for the first inspection, it was pretty intense though. And the inspector actually went and looked at each step, and our validation is complete for the entire process, DNA extraction, library preparation, sequencing, and analysis. And so the inspector went through each step, and he seemed to be pretty knowledgeable in our case. So he basically went and also looked at the DNA extraction kit box to see whether we had everything labeled and or not, and whether the QC was in place. And then he went to each room, each stations for the where the work is being done, and he looked at whether the instruments had tag for the preventative maintenance, and then he had questions about internal PTs that we put in place and competencies. He even went to the extent that the snip calling pipeline, he wanted to see whether our versions are locked. Like if we change version, then we have to do a mini validation in that case. So in our case, it was pretty simple because our Macs and computers are not connected to internet. So by accidentally, there will be no installation or upgrades. So that was pretty easy to defend. And then we showed the README files, which said which version we are using. And yeah, so he basically checked every process. And in 2016, that was our first inspection, and we were not doing external controls in every run. So that was our citation, that they said that we have to do external control for both DNA extraction and sequencing process. So that was a big citation. And going forward, like from 2016 to 2018, basically 2 years, we had to run a positive and negative control in every sequencing run. And you can imagine how much that costs. In 2018, then we put together our IQCP plan. And so since then, we have been sequencing positive and negative control only in every quarter. So in 2020, our inspector approved our IQCP plan. So finally, we got rid of not sequencing in every run. Yeah, I guess that's all our experience from California. Thanks.
Thank you, Rachel. And just to note, we organically went into the second question about your overall experience from CLIA inspection of next generation sequencing. So if any of the panelists would like to add on top of what you already said, any information about overall experience from the inspection, not related to validation and citations, please do.
Yeah, so Nick, this is Nick from Maine again. No, so our inspector looked at our processes for 4 or 5 hours, spent most of, we're a small lab, so she only ever comes from maybe 2 days max. And the second day was almost all whole genome. And she, once we explained each individual step, and sometimes we had to draw it out on a board or explain it again and again, they didn't find any citations. It was actually really cool. Most of the questions were around, well, why aren't you running controls? And it took so long. It's like, well, if I run an extraction control and it doesn't, I'm still doing a qubit reading to prove that I have, nucleic acid. So if it doesn't work, I'm just going to re-extract. So adding control doesn't add anything. Or if I'm adding like a species control, but I already know the species, so I don't need a species control if I'm just doing Salmonella serology or E. coli serology. And then down the line, it's like, well, I know Salmonella is going to have some sort of serological result, and same with E. coli. And if I don't have enough coverage and I don't get a serology, answer, I'm just going to resequence. And it might have to be that I have to re-extract. And we just went step by step by step to show that, why we didn't need controls.
They agreed with that you don't need controls?
Yeah, we had the same experience as Nick. We literally had to say, well, so if we have extraction and we're doing a positive and negative control, what does the positive control tell us? What if that failed, but we quantify the other 11 on the run and they all worked? And they couldn't answer that question, right?
Yeah.
We do of course, a negative control on our extractions to make sure we don't have contaminated reagents, but a positive control we didn't see necessary, and we were able to convince our CLIA inspectors of that. And it was the same kind of argument that Nick made, is that we're doing these continual QC checks throughout the process, right? We're extracting nucleic acid, we're checking to see presence of absence of that. We're going into library prep, we're then checking for the presence of absence, right, of an amplified library. And then when we do our QA QC check for our analysis pipeline, the first thing we do is the species determination. And if that's not what we thought it was supposed to be, or it's mixed or whatever else, that sample gets rejected and we go back to re-extraction. So, with those QC checks in place and with being able to sit, actually explain to the inspectors exactly what each one does, because a lot of them didn't have a lot of molecular biology experience, right? Like you had to really teach them that, when you're doing and prepping maybe 30 samples at once for NGS, that each individual sample is its own reaction set that's independent of all the other reactions. And they didn't quite understand that. They thought it was more like a multiplex PCR than it was actual individual single samples. But yeah, for us, the bigger thing was proficiencies and competencies. They wanted to make sure that we're getting our 2 proficiencies in, that we're doing our initial trainings and competencies and machine comparisons and kind of the things I think they were more comfortable with in terms of knowledge base. But yeah, that's where we were at.
Anybody else has experience with showing your controls to the CLIA inspector and being able or not to convince them that you don't need to do external controls? How did it go down?
Hey, this is Ritu again. Yeah, no, we tried to give a lot of arguments and evidence that we do not need positive controls. As Kelly mentioned, the fact is that we know the species, and so in the ID field, if we don't get it, then we don't need to proceed further. But yeah, unfortunately for us, it was, they did not. It went on for like 2 or 3 hours, but still they were very sure that they wanted us to do external positive and negative control too. So yeah, that did not work for us.
And actually, I had a similar question. We had CLIA inspection just about a few weeks ago, and I've asked the CLIA inspector about what's the position of the CLIA, if there's any updates on next generation sequencing, if there's any position from CLIA on the in silico controls available at this point. He had to go back and consult with his other colleagues, and the answer that I got that no, they still would like you to follow the general regulations. He ended up just sending me my own paper back in the end. So it just sounds that like they really pull out the regulations. A lot of them do, and this code just cannot go without me citing CLIA. But the CLIA says that the control procedures, first of all, they must detect the immediate errors, and that they must monitor the accuracy and precision of the complete analytic process. And then, let me see, where do I have this note? And then also additionally, specifically, it says that each qualitative procedure must include negative and positive control material. And there is actually a specific statement for the test, which has molecular amplification procedure. Those must include 2 control materials. So pretty much when they go off that regulation, I feel like sometimes even if they might even agree with you, that sure, it kind of makes sense that you have very stable and robust system. In the end, that's not what the regulation says. And that's where they can still get you, no matter how good your system is sometimes.
Yeah,
but I agree.
I think I'll add into that if I can. I think Nick had the right answer though. If you write in an IQCP, you get around that requirement for positive and negative controls with every run. You just have to explain it all, and the IQCP rules are a little bit of a pain. But I think with sequencing, you almost have to go down that road because that's what makes sense.
Absolutely, yes. Cannot agree more. And Ritu can talk about it more, and that's what California lab exactly did. They IQCP'd the controls as well. The only problem that with the QCP, you have to run the positive and negative control for a certain period of time before you can actually prove that it's working and there is no risk of getting incorrect results. So unfortunately, it's not the step you can skip completely and just say that, oh, we have all these other QC checkpoints in place and it will be fine. Unfortunately, again, it doesn't make sense, but oh well.
Yeah, I think in my instance, it helped that I was able to point to specific instances where controls would be absolutely required. If you're doing cancer genetics, or if you're doing, Huntington's genetics, you want a control there. You want to know how many times that, region has been duplicated. Or if you're doing metagenomics, you want to control, you want to make sure that you did extract something at the beginning, that you are amplifying something, because you might actually not have anything. But in the case of salmonella, if I'm taking a pure culture of salmonella, which I already know is salmonella, because I've already reported it out, and I'm extracting it, I know the extraction works because I'm running it on a qubit to get an OD. Unless that procedure is wrong, I'm going to agree that my OD is whatever, and then I'm going to dilute it down to give me whatever nanomolar or nanogram quantity I need. So now I know how much I have that I'm adding into my, my library prep. And then I know my library prep worked because on the machine, I can watch it, increase over time. I can watch my, my Q scores go. I can check all these other metrics that you predefine in your quality manual before. And then on the back end, once you start doing your in silico analysis, I know it's salmonella because I've already reported out salmonella. And I'm also going to do some sort of maybe metagenomic check just to make sure there's no contamination. And you say, and that particular database will be different between each individual laboratory. We don't run a lot of TB, so my metagenomic database does not contain TB, but it contains, like all the pulse net organisms and a bunch of HI organisms. And we've caught stuff on that end. You know, we had something that it looked great, and all of a sudden we had, I don't know how many, over like 1,500 contigs, and we had a mixed culture of like Klebsiella and, I don't know, I'm going to make this Klebsiella and E. coli for some reason. So they had to go back and reselect for each individual ones, and we were able to separate them out and reported out E. coli. So you have to specify each individual step and say, a control at this step doesn't work because my control is actually an OD reading, or my control is actually a Q score, or my control is actually how many contigs I need for this organism or that organism. Otherwise, you're saying, okay, if I want to prove Salmonella serology is working, I need to run Salmonella every time I have a Salmonella run. But for a small lab, my Salmonella run doesn't have just Salmonella. I'm going to have Salmonella and E. coli and Listeria because I don't have a lot of just Salmonella. So all of a sudden, I told our inspector, so my run can only have, say, 15, different types of samples, and now I'm running what I know are 7 different genus and species. So now I have 7 different genus and species controls. So I'm actually only looking at 7 samples. So when you break it down to each individual step, our CLIA inspector, we're due any time for our inspection, but we'll see how she thinks about it this time. And if you read, yeah, and I know they just opened up they closed it, maybe a year ago or so, that you could, as they're updating the CLIA regs, specifically for NGS, there was a big public comment period. And if you read those, that was one of the major concerns, is this need to do positive, negative controls when you have all these other different procedures or protocols in place to prove positivity and negativity along the way.
Absolutely, makes sense. Anybody else would like to comment on that?
Do what Nick did. It makes the most sense.
We tried. Oh, well. I just want to add on for the external controls. Nick, you were saying that, if you have 7 different things on your runs and you're a smaller lab or might only have a sequencer, like a low throughput sequencer or something, and that would be half of your space. We are able to rotate our controls. So we only have 1 positive control on a run, and it might be a mixed run. It might have organisms that would only be going for A and I identification, and it might have a handful of E. coli and a handful of Salmonella that would go on to their respective genotypers. We would only be required by CLIA to have one positive control and 1 negative control on that run. And we just rotate through them to ensure that we have coverage across all of our tests and all of our targets.
Yep.
And then if there's any sort of question about, say you have an E. coli and it just happens to be one of those targets where the coverage is right at the threshold, and so you really want to be careful about the call. We have the ability to import control sequences, so that you can do the analysis on that control sequence to ensure that it picks up as it should, and then assuming that can add to the assurance that call is accurate. It's sort of a safeguard. If you were uncomfortable about not having, if your control happened to be a Salmonella, but you were uncomfortable about doing the Escherichia analysis, we would have the option to do that. And I think I've heard of a couple of other folks who are using controlled assemblies or controlled sequence files to do this kind of a QC check in the on the dry side.
Actually, I have a follow-up question on that. For those labs who are doing positive and negative external controls or did at any point, is your negative control, is it a non-template control, or is it more like what CDC has in their toolkit? It is a non-target organism that is absent in your database. And just to now experience, CLIA inspectors often prefer to have non-template control as the negative control if there is an amplification step, because that's actually what controls for the contamination. And what would be your justification to prefer one type of the negative control over the other one? What's your experience? Anyone?
Can you say that one more time? I think I missed part of that. I'm sorry. I've got a
just like what you prefer, non-template control or the actual organism negative control?
Oh, I see.
Like a non-target organism.
I mean, I think it depends on what you're trying to control for, right? If you're trying to control for like reagent contamination, something you might see across the entire run, then a no template control makes sense. If you're looking to control for, you know, determination of a ANI value or something between two different species, then you might want to do a negative control that's a different species, right? It depends on what you're trying to control for.
And in this case, would it make sense, since we're talking about external controls, do you think it makes sense to actually sequence organisms that is your not target, or instead of using an in silico non-target?
No, I wouldn't. I don't think it makes any sense.
Okay, I agree, but I'm a moderator, I'm not supposed to say.
Yeah, no, I don't think you need to worry about that, because I mean, you can determine those things in silico, right?
Makes sense.
Yeah, I know when we do our metagenomics analysis, we have a database of we ran a whole bunch of negative, just nothing, just blanks through our entire process to figure out all the contaminating sequences in all of our reagents. And so we have a database of that. So that has helped a lot in that. Positive control, again, there is just, phi-X and MS2. You're just for us, it's just looking at virus. We're not doing bacteria. And then when we have our own internal viral database, we pulled out anything that's not human, that's not going to infect humans. So I don't care about tobacco mosaic virus. I don't. So pulled that out. There were a couple other things that we found in every single sample we ran. Torquetenovirus is in every single person, so I just pulled it out of the database because there's no sense in looking at it. And then, anything else that we found that was, that looked weird, but was, could be something from a virus that could infect a person, but they were using it. We had to systematically go through and say, okay, why am I seeing this sequence? Oh, this is a promoter region from one of the reagents. So I just have to make, I note that in our quality manual that if I see this sequence just a couple times, I'm not going to use it to call anything because I already know it's a promoter region, or it's, something else that they used, in a plasmid to make, Taq polymerase or something.
No, that makes sense, yeah. And I think we also settled with the non-template control because our justification was that, there is, what you're really controlling, what you should control is the contamination. That's the most problem which you most likely going to have with URSA. And the three most common windows where the contamination would happen is the DNA extraction, which could be reagents, but honestly, most likely it's cross-contamination. It's library prep before barcoding, or it is the contamination with the amplicons after the barcoding. And for the first two steps, our justification, the non-template control actually would control for contamination that occurs during DNA extraction or the library prep. But for that, you can't stop at the QC level because, for example, with DNA extraction, if you had contamination in your reagent, it could be a low-level contamination, which will show up on the sequencing, but you're not going to see it as a DNA concentration in your negative control. So that things need to be sequenced. And then, of course, with the library prep, there is potential that the contamination will also show up on the sequencing step, but you might not see it during your library QC. But then, of course, there's another consideration that we all probably have by now the contamination screen. So if your sequences are contaminated, you're likely to see the contamination screen flagging them, but that's more likely to happen if you have different species contamination. If it's the same species contamination, you might miss that. And that's why, per se, maybe drop in the non-template control in the beginning of validation before you get the IQCP. That's not something that we could justify internally. But then additionally, we control for the contamination with the amplified libraries later by using the kind of calling the indices that were used on the MASIC sequencer before, in the run before, but not in the current run, and see if there's any contamination with the amplified fragments from previous library prep. That, of course, doesn't control for if, let's say, there is a contamination with the library fragments with the same index combination as your current samples. But our thought was that this type of amplicon contamination is most likely to happen right after the run. So hopefully it will be controlled by the run that followed the previous run, because we do rotate indices. So that's where we start out with our negative controls. I don't know if anybody using that type of control for the amplicon contamination. That I would love to hear that, or what your thoughts about it.
I think we would like to hear from anyone on the call, not just a panelist who is doing this, right, Bavara?
Absolutely, yes.
Open to all.
There's a lot of chatter in the chat box.
Is there?
Yeah. Well, I guess there's a lot of conversation going on about SARS sequencing validation. I think maybe we just move to that topic at this point. And yeah, there's a lot of talking going on. If anybody would like to share, have you performed the CLIA validation for your SARS-CoV-2 sequencing? And are you reporting the SARS-CoV-2 variants at this point?
So we've validated the diagnostic test for presence or absence of SARS-CoV-2 in a sample, but we have not yet done it for variants. We're in the process of figuring all that out and the best way to go about it here. So we kind of have two parallel processes in place for sequencing of SARS-CoV-2. We have the COVID-seq kit and platform from Illumina, but we also have our hybrid Arctic/Illumina prep as well. And so, yeah, and I talked to our CLIA inspectors about this yesterday, and they have no answers. They really have no guidance whatsoever right now on the best ways to approach it, the best ways to validate it, anything. In fact, they said for anybody that's interested, they should email their regional CLIA inspector, and they can forward it up the chain of command to the central office, because they have no good answers. Because it's going to be very difficult, I think, to validate, right? With no gold standard in place, with no control materials, external control materials out there, it's going to be a lot of work for labs to really validate a variant call without doing a lot of work. So right now, all we're doing is reporting variants back to our epidemiologists, and that's it. We're not giving it back to clinicians, not getting into patient records at all. It's only going to epidemiologists. We'd like to get that hopefully under CLIA, but it's how we're going to do that. I haven't had a chance to sit down and think about. We're probably going to have to do it under our CLIA validated SARS-CoV-seq pipeline, right? Because every step along that way, from extraction all the way through analysis, has been validated. We'll just then have to add another validation on for variant determination. That's kind of how I'm thinking, but how to do that to the level we need to for CLIA is where I'm struggling.
Hi, this is Christina from Alameda County, and just to kind of follow up on what Kelly was mentioning, if there's no intent to kind of provide the results back to the patient or clinician, and it's mainly for surveillance purposes, there is no requirement for the validation, right? Like you could just be able to set up the sequencing, and if you just report back the results to your epi or public health department, that would
correct,
right? Okay.
I want to add a couple of thoughts on top of that. So Christina, I think you are about right. However, our current practice is once we find any significant variant of concern, that our epi will do a re-interview, basically to investigate, is there any further point of contact? Have you traveled or any of that? And oftentimes that the patient will ask, like, why you are interviewing me? And right now, our epi can mud it through. We do some further subtyping and blah, blah, blah. And if the patient further asks, like, oh, what kind of variant I get, then our epi is in this legal muddy water in terms of, do we tell them? Do we not tell them? And also, there might be some indication that the clinicians may choose to use or not use monoclonal antibody therapy if it's a B1351 or a P1 variant. So that is more of a direct hit in terms of CLIA validation.
Yeah, I think Sean's right. There are applications to having it validated, right? I think it's not a bad thing to have it done. I think there is some utility in validating it. It's just going to be not something you can do quickly, right? You're going to have to really think about, well, how are you going to control for 3 database updates in one day, right? How are you going to control for, you have no other validated test out there to tell you what the variants are, right? You have no control material to validate against. So you have to find ways to go around that. And I'm not sure how to do that right now. That's where I'm coming from. And yeah, we're kind of thinking, in terms of what Sean was saying about, not using monoclonal antibody treatments for patients is, we've looped in our clinical partners with the surveillance data, right? So if they see that P1 or B1.351 are circulating a lot in the cities, in the areas where these patients are from, they may choose not to do that. They may choose not to treat with monoclonal antibodies. It's not going into a patient record saying this patient has P1 or this patient has, B1.351, but it's really predominant in the area.
Yeah, I would just say, Jake Lemieux here from MGH in the Broad. I'm an ID physician and also interested in sequencing, and I can just attest that I was on discussion with hospital leadership today about what is the prevalence of E484K in our region. Some of the existing monoclonal products retain activity in the presence of that mutation, others do not. So this discussion is happening at in hospital systems across the country, kind of in real time. And this is from clinicians who are certainly not microbiologists. This is kind of hospital clinical leadership. So these are really happening fast, and there's a lot of interest in this area.
So this kind of makes me wonder then, like, given the difficulty in validating and the turnaround time associated with sequencing, why is a qPCR test for a specific mutation not a better approach?
I think it probably is. And I know one of our big clinical labs here in the state is working on developing a test that'll look for those things. It'll be a quick reflux from the positive, TAC path assay or whatever to one of those things. Yeah, it's definitely in process too, I think. It's just that also takes time to get validated and under their, CLIA regulations, the LDT and all of that kind of stuff. So they're working on it. And I even know Thermo Fisher is working on adapting the TAC path assay to have, a similar one for variants, to detect variants alongside the multiple assay.
So on top of that, I mean, yes, I was asked about this question as well. And sometimes my answer is, what about the unknowns, right? Like, just let's say 6 months down the road, you have another 10 variant of concern. Are you gonna be develop 10 different RT-PCR assay to detect that? Or, actually, I think actually that lead to a good sort of like discussion point. What should be the end product of the testing? Should it be the Pango lineage information, which I don't think is a good idea at the moment. You have the software was essentially being updated twice a day. or it should just emphasize on the key mutations, the E484K, the NALI, and other key mutations at the spike protein. So I think this is where, you know, things are certainly on, no one has figured this out yet, I think.
So before we continue on with the discussion, which is great, and I'm loving this so far, I do want to remind everybody that we are at the top of the hour, so I'm going to stop the recording for this hour. And I have the next hour blocked off to continue this discussion because I figured that there would probably be a lot of back and forth and probably a lot of interest. So if anybody is able to stay on the call, please do. We'll record for the next hour so that we can post that too for anybody who is not able to stay on. But I just want to be cognizant of everybody's time and say at least thank you so far to our panelists. I think that this was a great discussion so far, and you know, there's a lot here to unpack and a lot more work that is needed, I think, in this area. But just wanted to say thank you, everyone, for being on the call so far. So I'm going to stop the recording.